Hello everyone, I welcome you all on the SWAYAM course on genetic engineering and its recent advancement. I, Dr. Sharmisha Banerjee, working as an assistant professor in biomedical engineering and bioinformatics department, Chhattisgarh Swami Vivekanand Technical University, Bhilai, feel privileged to take you to this 30-minute journey into the fascinating world of gene therapy. Today, we will embark on a captivating exploration of this groundbreaking medical innovation that holds the potential to revolutionize the way we treat genetic disorders and various other diseases. Let's begin our journey by understanding what gene therapy is. Gene therapy, in other words, I would say it is a medicinal therapy or it is genetic medicine. Gene therapy is a cutting edge medical approach that involves altering or replacing the defective genes within an individual's cells to treat or prevent several diseases. It is like editing the software of life. So, before going into detail, basically we will quickly brush up what genes are. Genes are carried on a chromosome. It's a basic unit of heredity. It encodes how the protein is being made, or we can say it is from the central dogma of life. That is, we all know from DNA, RNA is produced and these RNA they make the proteins, and these protein they carry out most of the life's functions. When the gene gets altered, it causes dysfunction in the protein. Let's elaborate this. When there is a mutation in a gene, then it will change the codon, which will change which amino acid is called for which, and in turn it will change the conformation of the protein, which will ultimately change its function. So, genetic disorders results from the mutations within these genes, or more elaborately we can say it arises within the mutations in the genome. Human gene therapy seeks to modify or manipulate the expression of a gene or to alter the biological properties of a living cells, which are then further used for therapeutic purposes. Gene therapy is a technique that modifies a person's gene to treat or cure many diseases or disorders. It is a technique for correcting the defective genes that are responsible for the development of any disorder or disease, and basically there are four major approaches for the same. The first one is a normal gene is inserted to compensate for a non-functional or a dysfunctional gene. Second approach is an abnormal gene will be traded for a normal gene. That is, the abnormal gene would be swapped by a phenomena known as homologous recombination with a normal gene. Third approach is an abnormal gene is repaired through the process known as selective reverse mutation, which would ultimately reverse the mutation to a normal function of a gene. The fourth approach is changing the regulation of gene pairs, that is, it will control the expression of gene, and the mechanism will be similar to epistasis, when one gene affects the expression of the another gene. History. Let's look into the history of this gene therapy. Gene therapy has come a long way since its inception. It was first conceptualized in the year 1990s and has been significant advancements over the decades. In the 1980s, scientists began to look into the gene therapy. They have inserted the human genes into a bacterial cell. Then these bacterial cell had transcribed and translated the information into a protein. Then it was further introduced into the human cells. These proteins were further introduced into the human cells. In the late 1990s as well as early 2000, clinical trials began, marking the importance of milestone in the field. The first case of gene therapy was performed on September 14, 1990. The person was Ashanti De Silva, was treated for SCID, which is basically a form of severe combined immunodeficiency disorder. Doctors removed her white blood cells, inserted the missing gene into the WBC, and then put these altered cells or altered WBC back into the bloodstream of Ashanti De Silva. This strengthened her immune system, but though it succeeded, but the failure was that it only worked for a few months. There are several types of gene therapy, including gene replacement therapy, gene editing and gene silencing. In the first one, gene replacement therapy, from the name itself, the gene will be replaced. That is, it involves inserting a functional copy of a gene into the cells to replace the defective one. The second type of therapy is editing. Here, techniques like CRISPR-Cas9 are used, which allows the precise modification of genes to correct the mutation or introduce the therapeutic changes. The third one is silencing. This method aims to suppress the activity of harmful genes using techniques like RNA interference, which we have already studied in details in the previous lectures, and CRISPR-Cas9 we will study in subsequent lectures under genome editing techniques. Now the question is how the gene therapy works. A vector delivers the therapeutic gene into the patient's target cell. The target cells becomes infected with the viral vector. The vector's genetic material is inserted into the target cell. Functional proteins are created from the therapeutic gene, causing the cell to return to its normal state. Gene therapy works by altering the genetic code to recover the function of critical proteins. Proteins are the workhorses of the cells and the structural basis of the body's tissues. The instructions for making proteins are carried in a person's genetic code or variants or mutants. In this code, we can impact the production or function of a protein that may be critical to how the body works. Fixing or compensating for disease curing genetic changes may recover the role of these important proteins and allow the body to function as expected. Gene therapy can compensate for genetic alterations in a couple different ways. Gene transfer therapy introduces new genetic material into the cells. If an altered gene causes a necessary protein to be faulty or missing, then the gene transfer therapy can introduce a normal copy of the gene to recover the function of the protein. Alternatively, the therapy can introduce a different gene that provides instructions for a protein that helps the cell function normally despite the genetic alterations or mutations, we can say. Genome editing is a newer technique that may potentially be used for gene therapy. Instead of adding new genetic material, here editing is done. Genome editing is done, which introduces the gene editing tools that can change the existing DNA in the cell. Genome editing technologies allow the genetic material to be added, removed or altered at specific or precise locations in the genome. CRISPR-Cas9 is a well-known type of genome editing. Now the question is how this faulty gene is fixed or how the missing gene is fixed in gene therapy. Here we will study with the help of an example. As we can see in the figure that on the left hand side we have a viral genome. Okay. So, A, B and C, these are the three genes which are present on a viral genome, and out of these three genes, suppose A is the beneficial gene. So, what we are actually doing? The beneficial gene is being inserted into the virus and the gene which is responsible for causing infection or causing pathogenicity is basically removed from the viral genome, and in this way it will be a virus, it will be acting as a vehicle, it will be acting as a vector for incorporating this beneficial gene into the host cell where the alteration is required, or into the host cell whose original gene is non-functional or missing or dysfunctional. So, this viruses which will be used as a vector, it will be called as modified virus. Why? Here let us see with the help of elaboration. A virus is found which replicates by inserting its gene into the host cell genome. The virus has three genes, already I had discussed A, B and C. Now here gene A, it encodes a protein which allows the viruses to insert itself into the host genome. Gene B and C actually cause the disease this virus is associated with. I already told you that is the pathogenicity or the infectious gene segments which are responsible for causing diseases or infections in the host cell. So, the B and C gene will be replaced with a beneficial gene. Thus, the modified virus could introduce your good gene into the host cell's genome without causing any disease. So, we will use this modified virus to fix the broken window. The same is depicted here in the figure. Here we can see in the first segment or the which indicates the one segment of the figure. Here the healthy cells, we are incorporating the beneficial gene from the healthy cells into the viruses. Then these modified viruses are allowed to infect, I will say it infect, but actually the infection will not take place, but these unhealthy cells will recognize it as virus. It cannot distinguish because the coat material or the latching mechanism, everything remains the same, which the original viruses uses for infection purpose. So, actually we are cheating the cells or we are cheating the host so that these viruses gets incorporated into the host cell, but the genome is modified. So, what will happen? The beneficial gene will be incorporated into the healthy cells, the normal functions will be replaced in your unhealthy cells without causing any infection. So, this is how the viruses are used as a vector or as a mode of delivering our gene of interest into the host cells, and in this case beneficial gene is being introduced into the host cell. Viral vectors, the mechanism already it is clear, they replicate by inserting their DNA into a host cell. It can be RNA in some cases. Gene therapy can use this insert gene that encode for a desired protein to create the desired trait, and the viral vectors which are more commonly used for gene therapy belongs to 4 different types. They are, the first one is retroviruses. They create double stranded DNA copies from the RNA genome. The retrovirus goes through reverse transcription using reverse transcriptase and RNA. The double stranded viral genome integrates into the human genome using integrase. These integrase, they are the enzymes which inserts the gene anywhere because it has no specific site. Now, this is the major caution with these types of vectors. It may cause insertional mutagenesis and it is not desirable. That is, one gene may disrupt the another gene's code, or the disrupted cell division can cause cancer from uncontrolled cell division. Vectors used are derived from the human immunodeficiency viruses, that is HIV, and they are being evaluated for safety purpose beforehand itself before being used for the vector purposes. The second category of the viral vectors are adenoviruses. They are again the double stranded DNA genome that causes respiratory, intestinal and eye infection in humans. The inserted DNA is not incorporated into the genome, it is not replicated through, has to be reinserted when more cells divide. So, the most common example of such type of viruses is common cold viruses. So, it is for the short time. When the cell divides again, these type of viral vectors needs to be used again and again. So, this is the limitation of these type of vectors. This is a figure which shows, on the left hand side we can see how the viruses, the genome of the viruses can be DNA, RNA as well as protein. So, here we can see how the viruses genome is being modified. So, the blue part indicates the original genome of the virus, and in red we have inserted our gene of interest which needs to be replaced or which needs to be cured. So, what is happening? In the first step we can see the virus is latching on the cell. It is then incorporated into a vesicle. This vesicle then ruptures and fuses with the cell membrane, and it will incorporate our gene of interest into the host cell, and the protein which was being malfunctioned from that gene is again reverted back to its original state. Whereas in the upper right side we can see the enlarged view of how the cell is being lashed with a virus or with a virus vector, we can say. The third category is adeno associated viruses. The adeno associated viruses, they are small single stranded DNA that inserts the genetic material at a specific point on chromosome 19. It belongs to parvovirus family. It causes no known diseases, hence it is a most common choice of vector and doesn't triggers the patient's immune response. It has low information capacity. The major drawback here is the gene is always in the turned on state and the protein is always being expressed constantly, possibly even also in circumstances or instances when it is not needed. It is basically used in hemophilia treatments. For example, a gene carrying vector could be injected into a muscle, prompting the muscle cells to produce factor 9, and this helps in prevention of bleeding. It was studied by Wilson and Cathy from University of Pennsylvania that these patients, they do not require factor 9 injections for more than a year, means this treatment was cured, and within a year the factor 9 used to be produced inside the host cell, we can see. So, this was a very remarkable achievement that when adeno associated viruses were used as vector for hemophilia treatment. The last category of the virus vectors are herpes simplex virus. The mechanism remains more or less same for all these 4 type of vectors, but the structure varies slightly and their latching mechanism will differ, and the genome which they carries is also different in all of these virus vectors. Here it is again a double stranded DNA viruses that infects the neurons, and the example of this is HSV. In short, it is known as HSV, herpes simplex virus type 1, and the right side of the figure indicates the structure of the same. Gene therapy with viral vectors has been successful, but it does carry some risk. Already I had discussed few of them. Sometimes the virus triggers a dangerous immune response. In addition to this, vectors that integrate the genetic material in a chromosome can cause errors because the integration site is not specific, it is not precise. So, it can even lead to insertional mutagenesis, or it can even lead to disastrous conditions like cancer. Hence, it led to the exploration of non-viral modes of gene therapy. Non-viral options can be categorized into 3 major groups. The first one is directly introducing the therapeutic DNA, but the limitation is it can only be done with only certain tissues. It requires ample amount of DNA, it requires a large amount of DNA. The second option is creation of artificial lipid sphere with aqueous core, which is basically known as liposome. It carries the therapeutic DNA through membranes. The third category of non-viral options is chemically linking the DNA to molecule that will bind to special cell receptors. DNA is engulfed by the cell membrane, but the technique is less effective. Researchers are developing newer technologies that can deliver the genetic material or gene editing tools without using viruses. One such technique uses special structures called nanoparticles as vectors to deliver the genetic material or gene editing components into the cells. Nanoparticles are incredibly small structures. From the name itself, they are of the size range of within the 100 nanometer. They have been developed for many uses. For gene therapy, these tiny particles, they are designed with specific characteristics so that they can target them to particular cell types, means they will act as a ligand, and these specific cell types have some sort of particular receptors on their surface. So again it is basically like a lock and key mechanism. We know that a lock gets opened only with a specific key. Similarly, this specially tailored or designed nanoparticles will only latch onto these specific cell types because of their corresponding receptors on the surfaces of those cells. They are less likely to cause immune response with as compared to the viral vectors, as they are easier to design and modify for specific purposes. Researchers continue to work to overcome the many technical challenges of gene therapy. For example, scientists are finding the better way to deliver gene or gene editing tools and target them to particular cells. Here I will discuss the unsuccessful case study of a gene therapy. Patient named Jesse had suffering from ornithine transcarbamylase enzyme activity and died in the year 1990. Within hours after the doctor shot the normal OTC gene attached to therapeutic viruses into his liver, Jesse developed high fever. His immune system began raging out of control, blood began to clot and the immune system failed, and in this way the patient could not survive, the lung shut down and the patient could not survive. So, the problem with gene therapy was that it does not have control over the gene where it will be inserted into the genome. The location was not precise, hence it was unsuccessful. Successful 1-year gene therapy trial was done for the Parkinson's disease. The unsuccessful story was also important to discuss with you all, because the attempt was made, but because of the shortcomings it was not possible. So, Neurologics, it is a biotech company, announced that they have successfully completed the landmark phase 1 trial of gene therapy for a neurological disorder, that is Parkinson's disease. This was a 12 patient study with 4 patients in each of those dose existing cohorts. All procedures were performed under local anesthesia. Primary outcome of the study was safe and the tolerability were successfully met. So, there were no adverse event reported relating to the treatment. So, this was the successful therapy which was being quoted by Neurologics company at first. Other successful case studies where the gene transfer procedure utilized the AAV, that is adeno associated virus vector, a virus that has been used safely for a variety of clinical gene therapy trials. The second study was Luxturna. This gene therapy treated a rare form of inherited blindness and has reported the vision in some patients, restored the vision in some patients. The another story was CAR T cell therapy. It involves genetically modifying a patient's immune cell to target and destroy the cancer cells, leading to remission in some leukemia patients. The recent developments into the field are the genes get into the brain using liposomes coated in a polyethylene glycol for treating the Parkinson's disease, RNA interference or gene silencing to treat Huntington's disease was used, siRNA was used to degrade the RNA of particular sequence, abnormal protein could not be produced here. Created tiny liposomes that can carry the therapeutic DNA through pores of nuclear membrane, and the another development was sickle cell was successfully treated in mice. The clinical trial still needs to be done for humans. There are a variety of types of gene therapy products which includes plasmid DNA and viral vectors. So, circular DNA molecules can be genetically engineered to carry the therapeutic genes into the human cells. The second product was viral vectors. Viruses have a natural ability, already we have seen, to deliver the genetic material into the cells, and therefore, some gene therapy products are derived from viruses. Once the viruses have been modified to remove their ability to cause infectious disease, these modified viruses can be used as vectors or vehicles to carry the therapeutic gene into the human cells. Bacterial vectors, they can be modified to prevent them from causing infectious disease and then used as vectors to carry therapeutic genes into the human tissues. Human gene editing technology, the goal of this is to disrupt the harmful genes or to repair the mutated genes. The next product is patient derived cellular gene therapy products. Cells are removed from the patients, genetically modified using vectors and then returned back to the patients. These are an example of customized or personalized medicines. We have heard about the customized costumes, but right now we can see how far we are progressing, that we are working on customized medicines using gene therapy. So, these are the some diseases where gene therapy can be applied, and the right hand side of the table indicates the target cell where the gene therapy can be used. The first disease is severe combined immunodeficiency (SCID). The target cells are B lymphocytes or T lymphocytes. Second one is hemophilia. The cells are liver or muscle. Cystic fibrosis, it affects the lung cells. Cancer, it can be many or any cell types. The next one is neurological diseases or disorders like Parkinson's and Alzheimer's. Again, the defect is in the nerve cell. Infectious diseases like AIDS or hepatitis B, where the white blood cells are basically manipulated or the white blood cells carries the defect. Gene therapy could be very different for very different diseases. The first one is it can be gene transplantation for the patients where the gene deletion has occurred. The second one is gene correction, so as to revert this specific mutation for the gene of interest. The last one is gene augmentation, that is the expression of gene of interest is basically enhanced in this type of approach. This is a figure which shows how the gene therapy is classified into two different types. One is your ex vivo and the second one is in vivo. From the name itself, ex vivo means the cells are cultured outside, extracted from the patient, cultured outside and then again reinserted into the patient, whereas the in vivo it is completely done inside the body. So, these are the two examples. In detail we will study in the next slide what are the techniques involved in it. Coming to in vivo gene therapy, the genetic material in vivo means completely it will be done inside the patient. The genetic material is transferred directly into the body of the patient. It is more or less a random process. Here we cannot have manipulations, larger manipulations, and we have very lesser ability to control the process. So, the third one is it is the only available option for tissues that cannot be grown in vitro, because any tissues which cannot be grown in laboratory conditions or which cannot be cultured, they are basically treated with the help of in vivo gene therapy, but it is not preferable or advisable in cases where the ex vivo can be done. Ex vivo gene therapy, the genetic material is first transferred into the cells, it is grown in vitro, it is basically a controlled process. Genetically altered cells are selected and expanded and major manipulations can be done through this process. Cells are then returned back to the patient. Now, let us delve into the various applications of gene therapy. The first one is already we have discussed, it can be used for treating genetic disorders. It offers hope to individuals suffering from diseases like cystic fibrosis, sickle cell anemia and muscular dystrophy. Dystrophy means dysfunction. The second, it can be used for cancer. Immunogene therapy and oncolytic virotherapy are being explored to target and destroy the cancer cells. Inherited disorders like hemophilia and certain forms of blindness can also be targeted for treatment using gene therapy. Now, let us discuss the gene therapy for cystic fibrosis. It is an ideal candidate for gene therapy for four main reasons. They are, it is a single gene defect, it is a recessive condition with heterozygotes being phenotypically normal, the main pathology is in the lung, which is accessible for treatment. It is a progressive disease with a virtually normal phenotype at birth, and it offers a therapeutic window because of these reasons. As with any groundbreaking technologies, gene therapy also comes with challenges and ethical considerations. These includes concerns about safety, accessibility and the potential for unintended consequences. Ensuring equitable access to gene therapy is also a significant concern. There are certain problems with gene therapy. The first one is it's short lived. It's hard to rapidly integrate the therapeutic DNA into the genome, and rapidly dividing cells would have to be manipulated several rounds for therapy. Multiple rounds has to be used for therapy. Second is immune response. New things introduce the immune system to get stimulated or activated. The third one is viral vectors are challenging. Then the next one is multi-gene disorders cannot be treated using gene therapy, like heart disease, high blood pressure, arthritis and diabetes. They are hard to treat because we need to introduce more than one gene. So, at a single time, again it is a major challenge. I will not say it is not possible, but it is a major challenge. May in future some of you may explore this or may sort out for this, looking forward for the same. May induce a tumor if integrated in a tumor suppressor gene, because insertional mutagenesis can occur. What are the ethical issues surrounding the gene therapy? The first one is, who will decide, or how can the good and the bad users of these technologies be distinguished? Who will decide which traits are normal and which constitute a disability or disorder? Will the high cost of gene therapy make it available only to the wealthy people or richer segment of the society? Could the widespread use of gene therapy make society less accepting of people who are different? Should people be allowed to use gene therapy to enhance basic human traits such as height, intelligence or athletic ability? These are the future prospects of gene therapy. Researchers are working on improving the safety, increasing the efficiency and expanding the range of treatable diseases. Personalized medicines, where therapies are tailored into an individual's genetic makeup, is on the way. In conclusion, gene therapy is a groundbreaking medical field with the potential to transform the way we treat diseases. While there are challenges and ethical considerations, the success achieved so far and the ongoing research offer hope for a brighter and healthier future. Thank you for joining today. I hope you found this journey into gene therapy both enlightening and inspiring. The future of medicine is in our hands, and with continued research, collaborations and joint efforts, we can unlock the full potential of it. Thank you.